Okay, let's just mention the last important point related to the gold nanoparticles and then move into the new imaging techniques. As you see here, the gold nanoparticles can be detected under the transmission electron microscopy imaging. Okay, this technique uses the electrons. When the gold particles are present in the tissue or cells, they can be detected as the black spots here. And this good detection can be used to see where the nanoparticles are going, whether they have been internalized by the cells or not. And that's it. But now let's go into a new imaging technique which is highly used for the cancer therapy, and it is somehow a little bit complicated technique. This technique is used the positron emission tomography, known as the PET. Well, it uses the radioactive materials as the imaging modality, as the tracer to send the signals towards our detector. If you remember, we mentioned that in the case of fluorescence microscopy, these are the fluorophores which send the signals. In the case of MRI, there are the relaxivity of the hydrogen atoms in the water contained in the body, which we get the signal from, and only the presence of the magnetic nanoparticles will increase the contrast of the MR images that we can take. And that was the reason that we mentioned magnetic nanoparticles are not the tracers, they are the contrast agents. But here in this imaging technique, the radioactive materials, the radionuclides, they are used as the tracers, because when we introduce them into the body, they will emit two gamma photons, as you see here, one gamma photon here and one gamma photon here, and they have high energy, and they have no penetration depth limit, so they will travel out until they reach the detector, and after that the image will be processed by the computer to get the three-dimensional image of our body. The idea is that if we bind the radionuclides on some biologically active molecules such as the glucose, we can see whether the radionuclides have been internalized into the cancerous tumor or not, because the cancer needs the energy and it uses the glucose a lot. So by uptaking the glucose, also the radionuclides which are connected to our glucose will accumulate into the tumor, and as a matter of fact, we can get better localized positron emission tomography images of the cancerous tissues. However, there are two disadvantages related to this technique. The first one is the short lifetime of the radionuclides, and they are synthesized in a synchrotron. So we need to make sure that within that short life of the nuclides, we can go into the hospital and introduce them into the body of the patient, and etc. The second problem is that these nuclides have to be in the short distance of the tumor tissue. So we need to use a vehicle, something which guarantee that these nuclides are going into the tissue. For example, we need the glucose, we need some other nanoparticles to take them, deliver them into the tissue. Just remember that linking the nuclides onto these glucose or the vehicle or whatsoever needs a chemical reaction. So it would be time consuming, energy consuming, and so complicated. And as the last slide of all the information about imaging techniques, you see here that we have the polymeric nanoparticles which have the linkage on their surface, and this linkage can be used for the absorption of these nuclides, radionuclides, on the surface of these polymeric nanoparticles. And you see here, this will be the resultant nanoparticles, which are equipped with the positron emission tomography tracers. And here you can appreciate the pictures related to the images that we get. And as you see here, after about 120 minutes, after 2 hours, they are accumulated within the cancerous tumor. Here you see the tumor that have been grown on these mice, and they are accumulated there, and the PET images have been taken. Okay, all of these slides about the imaging techniques are finished, and I hope that you find them helpful. And I have to mention that these are the references, and if you want to get more deeply into the information in these slides, you can visit any of these that you like.